So my topics is all about the chapter 17, and it is the cell cycle. So the overview of the cell cycle. So the first one is the eukaryotic cycle usually consists of four phases. So it is eukaryotes that the cell cycle consists of four discrete phases. So the first is the G1, S, G2, and M. So the S or synthesis phase is when DNA replication occurs, and the M or mitosis phase is when the cell actually divides. So the other two phases, G1 and G2, the so-called gap phases, are less dramatic but equally important. So the next one is the cell cycle control is similar in all eukaryotic cells. It is the cell cycle and its system of checkpoint controls show strong evolutionary conservation. As a result, all eukaryotes from single-celled yeast to complex multicellular vertebrates pass through the same four phases and same K checkpoints. So the third one is the cell progression can be studied in various ways. So it is the cell cycle progression can be studied in various ways, and one way is to simply look at living cells with a microscope. It is a glance at a population of mammalian cells proliferating in culture reveals that a fraction of the cells have rounded up and are in mitosis. Others can be observed in the process of cytokinesis. So the next one is the cell cycle control system. So the first is the cell cycle control system triggers the major events of the cell cycle system. So it is the cell cycle control system operates independently of growth factors. So the cell cycle control system receives messages from outside and inside the cell, triggers and controls major events in the cell cycle, and includes 3K checkpoints to complete a cell cycle. So the next one is the cell cycle control system depends on cyclically activated cyclin-dependent protein kinases. So the cyclin-dependent kinases, or the CDKs, are the families of protein kinases first discovered for the role in regulating the cell cycle. They are also involved in regulating transcription or mRNA processing and the differentiation of nerve cells. So the next one is the regulated proteolysis triggers the metaphase to anaphase transition. So it is the APC dependent proteolysis triggers the separation of sister chromatids at the metaphase to anaphase transition and the destruction of mitotic cyclins at the end of mitosis. So the next one is the cell cycle also depends on transcriptional regulation. It is the internal and external factors regulate cell division. So it is external factors include physical and chemical signals. Growth factors are proteins that stimulate cell division. Most mammal cells form a single layer in a cultured dish and stop dividing once they touch other cells. So the next one is the cell cycle control system functions as a network of biochemical switches. So it is a series of biochemical switches control transition between and within the various phases of the cell cycle. So the cell cycle is a series of complex, ordered, sequential events that control how a single cell divides into two cells and involves several different phases. So the next is the S phase. So the first one is the is CDK initiates DNA replication once per cycle. So this is CDK phosphorylates proteins at the DNA replication origins, initiating DNA synthesis through a mechanism that ensures the DNA is duplicated only once per, per cell cycle. So the next one is the chromosomes duplication requires duplication of chromatin structure. So it is the duplications arise from an unequal crossing over that occurs during meiosis between misaligned homologous chromosomes. So the chance of it happening is a function of the degree of the sharing of um, repetitive elements between two chromosomes. And the last one is the cohescence holds sister chromatids together. So a protein complex called cohesin that is conserve one single-celled organisms all the way to humans is responsible for keeping the sister chromosomes together by forming a ring around the DNA. So despite the importance of this complex, exactly how it interacts with the DNA in the chromosomes was unknown. So the next is the mitosis. So this is this picture goes like this. So the first is the interphase, the prophase, inlaid prophase, metaphase, anaphase, telophase, and the cytokinesis daughter cells. So in cell biology, mitosis is a part of the cell cycle in which replicated chromosomes are separated into two new nuclei. Cell division gives rise to genetically identical cells in which the total number of chromosomes is maintained. Therefore, mitosis is also known as equational division. So next is the cytokinesis. The cytokinesis is the part of the cell division process during which the cytoplasm of a single eukaryotic cell divides into two daughter cells. So the two daughter cells, which is this one, the contractile ring creating cleavage furrow. So which is this one, the blue one, and the second one is the completed nuclear envelope surrounds the condensing chromosome. So it is a cytoplasmic division begins during or after the late stage of nuclear division in mitosis and meiosis. So the next one is the meiosis. Meiosis is a special type of cell division of germ cells in sexually reproducing organisms used to produce the gametes such as sperm or egg cells. It is involved two rounds or of division that ultimately result in four cells with only one copy of each chromosome. So the meiosis goes like this versus the interphase. Meiosis 1 homologous chromosomes, the daughter nuclei, and the daughter nuclei 1, and the meiosis 2. And the next is the control of cell division and cell growth. So the first is the mitogenes can stimulate cell division. So what is the mitogenes can stimulate cell division? So it is the cell division by increasing the amount of G1 cyclins. So the G1 cyclin CDK leads to active G1s or S cyclin CDK by increasing the transcription of G1 RS cyclin and removing an inhibitor of G1 RS cyclin CDK. Then it triggers activation of 53, I think, based on my research, 53 increases that amount of proteins that inhibit G1 or S cyclin CDK. So the next one is the many human cells have a built-in limitations on the number of time they can divide. So So the Hayflick limit is a concept that helps to explain the mechanisms behind cellular aging. So the concept states that a normal human cell can only replicate and divide 40 to 60 times before it cannot divide anymore and will break down by programmed cell death or apoptosis. So the next is the abnormal proliferation signals cause cell cycle arrest or apoptosis except in cancer cells. So there are many signals that can occur in cancer cells that quickly leads to apoptosis despite their typical evolution evasion of apoptosis. So cancer cells are primed for death, meaning that they are closer to triggering the apoptotic pathway than normal cells. So the next is the cell proliferation is accompanied by cell growth. So the normal proliferation occurs following a four-step cycle known as the cell cycle G1 phase, when cells grow one stack up on nutrients. Next is S phase, when their DNA is duplicated. The third one is the G2 phase, when DNA is checked for duplication errors, and M phase, the last one, when the cell divides, giving birth to two new cells. So the last one is the proliferating cells usually coordinates their growth and divisions. So it is most basic level. Cell size, homeostasis, and proliferating cells requires a coordination of growth with division such that on average each cell division is accompanied by a doubling in cell mass. So in post-mitotic cells such as neurons, the maintenance of cell size requires that no net cell growth occurs. So that's all about for my reporting about the cell cycle.